Hello, everybody, and welcome to Science Abounds Science at Home, where we will discover home lab magic. We will demonstrate experiments demonstrating scientific principles, concepts, and reactions in chemistry, biology, microbiology, microscopy, and molecular biology. Welcome to my channel. Hello, everybody, and welcome to another episode of Science Abounds Science at Home, where we will discover home lab magic. I'm very excited by today's episode. We are going to be looking at SDS-PAGE Protein Electrophoresis. SDS-PAGE, or polyacrylamide gel, is a laboratory technique used to separate proteins based on their molecular weight. It's one of the most common methods used in biochemistry and molecular biology for analyzing protein purity, estimating molecular size, and checking expression levels. Now, I've made a previous video on gel electrophoresis where we looked at the separation of some DNA sample. DNA analysis or DNA electrophoresis uses a horizontal setup and it uses agarose gel. For protein separation, we use a vertical electrophoresis chamber, and we use polyacrylamide gel instead of agarose. The reason why protein electrophoresis is usually run vertically is to do with the gel type, the buffer system, and the need for even migration and heat control. And in protein electrophoresis, we use polyacrylamide gel, or PAGE for short, which is cast between two glass plates. So I use pre-made gel plates. You can buy these from a variety of suppliers. You can check out my video description for all my suppliers where you can buy these pre-made acrylamide gels. But if you were making them yourself, you would have to pour the polyacrylamide gel between the two glass plates. And that's why it's easier to do it vertically and then casting it horizontally, which would be very difficult to do. Because polyacrylamide gels are also thinner than DNA agarose gels, and just for the interest of time and consistency and uniformity, I always buy them pre-made rather than casting myself, because it's quite challenging to do yourself. The gel is mechanically fragile and relatively thin, as I said, so it is easy to mount it vertically between the plates for support and to ensure polymerization and contact with the buffer. SDS, where does the SDS come from and what does it mean? SDS stands for sodium dodecyl sulfate, and it is a detergent that binds to proteins and helps denature them. Each protein comes coated with SDS molecules, giving it a uniform negative charge proportional to its length. When an electric field is applied across the polyacrylamide gel, all proteins migrate to the positive electrode, but their rate of movement depends on their size and molecular weight. Small proteins move faster through the gel than larger proteins. The objective of this video is to use SDS-PAGE electrophoresis to develop an understanding of protein structure, function, and diversity. We're going to be separating and analyzing several different types of protein which vary in different molecular sizes. And we're going to use the SDS-PAGE system in order to demonstrate how you can identify proteins in unknown samples based on their molecular weight and migration through the gel. So what kind of proteins are we going to be looking at? First, we're going to be looking at milk proteins. Milk contains 3 to 4% protein, divided mainly into casein and whey proteins. Casein exists as micelle spherical complexes, stabilized by potassium casein and calcium phosphate. Casein provides the main source of amino acids, calcium and phosphate, and is responsible for curd formation in cheese and yogurt. We're also going to look at blood serum proteins. These are proteins found in the liquid portion of blood after clotting has occurred. Serum consists of albumin, which is the most abundant serum protein, and maintains oncotic pressure and transports molecules such as hormones and drugs. You also have the alpha-1 and alpha-2 globulin proteins, which bind hemoglobin and transport copper. Then you have the beta globulin proteins, which are involved in ion transport and in immune defense. And finally, you have the gamma globulins, or immunoglobulins as they're known as, which are antibodies, and these are used in immune defense. We're also going to look at egg white proteins. And these are often called albumin. And albumin is a collection of proteins found in the clear part of an egg. These proteins perform protective, nutritional, and functional roles. And finally, we're going to look at plant proteins, which are storage proteins, which include storage proteins such as lugamin, vicillin, which are found in legumes, and then prolamines, which is found in cereals. Then another group of proteins found in plants are enzymes, such as peroxidase, which is important in metabolism and defense. And then you have structural proteins called extensins and defense proteins, such as trypsin inhibitors. So what we're going to do next is begin the experiment. We're going to prepare our tank. I already have buffer, the SDS-TRIS buffer already prepared. And then I'm going to load my gel and get that ready. So that's going to be step 1 of the experiment process. Okay, so the first step we're going to do is prepare the electrophoresis tank and load and place the gel. So I'm going to take the gel out of its package, slide it out like so. I'm going to take the lid off the chamber for a moment while I load the gel. What I'm going to do is I want to remove the comb that has created the wells, so I don't need that in there anymore. So I'm going to gently remove the comb out, trying not to disturb the wells, like so. Okay. And next is I'm going to place the gel into the tank, and it just slides in. And I highly, highly recommend for protein electrophoresis, if you're going to do this at home yourself, if you want to do this yourself, to buy pre-made gels. There's simply no better alternative. And next is I'm going to pour in the buffer. Now, sometimes the buffer, the SDS, can precipitate out and lead to a cloudy solution. And you really don't want that cloudiness. So if you gently heat the buffer, it will re-dissolve the detergent, the SDS, and you'll end up with a nice clear solution. So I want to put enough buffer to cover the gel and the wells. Okay. Let that equilibrate a little bit more and then pour in some more. Oops, I'm making a mess. Okay. All right, and then what I want to do is, once that's settled out, is I want to rinse out the wells with buffer. And I'll do that in just a moment. I will show you how to do that. So the next part of the experiment is, I'm just going to let the bubbles settle so you can see things better. The next part of the experiment is you want to deproteinize your protein samples. And you do that simply by boiling water and letting the samples sit in boiling water for about 5 minutes. So let's do that next. So I've placed my protein samples into a boiling beaker, a beaker with boiling water. So I'm going to let that incubate for about 5 minutes to denature the proteins and unravel, because the protein samples will not separate if they are not denatured. So we'll wait 5 minutes to incubate them and then apply them to the gels. Okay, so now I'm going to apply the samples to the wells, and I'm going to pipette about 30 mLs, 30 microliters of my protein sample to each well. And you want to apply your sample gently and slowly to give it enough time to settle and go and settle at the bottom of the well. Okay, so I've applied my samples and now it's time to perform the electrophoresis. I'm going to put the lid back on like so, and then I'm going to connect my leads, my electroleads to the power source. So I've connected my leads to the power source. I want to do the electrophoresis at 150 volts for 1 hour, so 60 minutes. So I'll set the timer on to 60 minutes and then start the electrophoresis. Okay, it's now running and we will come back when it's done in 60 minutes. Okay, the electrophoresis run is now complete. So we are going to move on to stopping the run and staining the gel so that we can visualize the separated bands. So first things first is to disconnect the cables from our power source and then remove the gel from the tank. Now, in order for us to stain the gel, we have to remove the gel from the 2 glass plates. And in order to do that, what I'm going to do is I'm going to use a spatula and I'm going to remove the top glass plate, leaving the gel on the back plate. So I can use a spatula to pry open the glass plates, trying to separate them. It is kind of fiddly to do and you don't want to damage the gel in any way. But eventually, because the plates are like clicked into place, so once you release them, it's fairly easy then to remove that top plate. Okay, so now I've prized away that top plate and removing it completely from the gel. If some of the gel sticks to that top plate, you can just simply, using the spatula, gently tear it away. Okay, so now what I want to do is I'm going to add some staining solution, which is made up of distilled white vinegar and ethanol. So I want to add a little bit to my pan and then I'm going to let that help in separating the gel from that back plate. So now what I want to do is remove the back plate, again being careful not to damage the gel in any way. And it should easily slide off like that. Okay. Now that's been removed from the glass plates, I can then move on to staining. And I'm using a fast blue stain. Again, it's made up in that solvent, the distilled white vinegar and ethanol. And I'm going to let that stain for a good 15-20 minutes. I'm just going to leave it in the pan to stain. Okay, it's been a good 20 minutes, so I'm going to siphon off the stain into my waste beaker. And then I'm going to add some of that destainer solvent. And so I rinse it in the destaining solvent and then leave it for about 15 minutes to destain. And then I pour that off and add fresh destainer and then leave it for a good 1 hour to 90 minutes depending on the gel, if it's how clear you can see the bands. So I'm gonna add some fresh destainer and leave it sitting for at least an hour to destain. So here is the results of our gel separation. If you remember, in lane 1, we had the standard protein markers showing relative positions of the protein bands with their known molecular weights. In order to determine the molecular weight in our samples, we can measure the migration distance in centimeters of each major band. All measurements should be from the bottom of the sample well to the bottom of the protein band. Using semi-log graph paper, you can plot the migration distance of each standard to produce a straight line. Using this standard curve, you can then determine the molecular weight of the unknown proteins in your samples. Well, I hope you enjoyed that video demonstrating the technique of SDS-PAGE protein electrophoresis. We'll come back and use that technique again in some future videos. Please, if you enjoyed the video, hit the subscribe button. It's free and it shows me your support, keeps me going, making more and more videos of a variety of different experiments. Please feel free to leave me a comment. I'd love to hear from you. You can also send me an email, send me your questions, comments, opinions, and also send me your photos and videos of you doing home lab experiments. Until next time, keep on experimenting. Goodbye, everybody.